Thank you, the organizers, for inviting me. I will speak about BAL gene expression as an indicator of outcome and disease mechanisms in IPF. So you already heard a bit about the pathogenesis of IPF. I just want to mention, and you know that the hallmark of IPF, of the UIP pattern, is the fibroblast foci. And we know quite for a while now that all these myofibroblast foci are connected, so they form a net, a mesh of fibroblast strands all over the IPF lung. And it's not a foci, as the name is telling. It is more a mesh of fibroblast strands, which was shown nicely by a pathologist in a 3D reconstruction, and her name was Caroline Kuhl. This fibroblast mesh is accompanied by a capillary mesh, shown here in this figure in red. The other hallmark of IPF is the honeycombing, and you know that we diagnose the UIP pattern on the CT scan by looking for this honeycombing. However, in the current scheme that we have, how IPF is evolving, the honeycomb changes are considered to be present only at the very end. So they indicate in this scheme that this is only present in very advanced disease, and a mechanism that is going on only at an end stage type of a disease. So we do not think that this is true, and we challenge this concept. So what we did is we studied bronchoalveolar lavage. So BAL is a widely used diagnostic tool in Europe still. So within our diagnostic routine program, we use it. So basically, more or less every patient get a BAL in Europe. And we used these cells to study the BAL-Transcriptome. In total, we included at the end the data sets of 176 patients in our analysis. We started with my own former cohort from Freiburg, Germany, and later added 2 other cohorts from Siena, Italy, and from Leuven, Belgium. So this project was done in collaboration with Naftali Kaminski and his group. And at that time, so we started that project in 2010, so quite a while ago. And at that time, we used the available system, the microarray, and an Agilent platform. So what we found is, we found a very strong, robust signature in the BAL, so mRNA transcripts that indicate mortality in IPF patients. So this is really fascinating. So it is a very robust signal that you get out of the BAL, and I think it is much more closer to the tissue, to the lung tissue, than to the blood. And we confirmed the signature, so our statistician built up a score, and he came up with a 9-gene signature out of these 1,582 genes or transcripts. And by using this 9-transcript signature, he could confirm that also in the other 2 cohorts, in the Leuven and in the Siena cohort, so this signature is performing very well and is predicting mortality in IPF patients. And when we looked at the genes, so we used software tool which can be used to predict upstream regulators of our signature, mechanism underlying this signature. And to our surprise, we found that the first hit is cancer, organism injury and abnormalities, and reproductive system disease. So later we learned that this is a stem cell signature or a stem cell indicator. when we started this project, in the beginning, we thought that we will find macrophage genes. You may know that I published on CCL18. We thought that we will find neutrophil genes because most of the cells that you will find in the BAL are 90% are macrophages, 10 around lymphocytes, and then there is a population of neutrophils. But we did not thought that this will reflecting a rather tissue-derived signature. So, and when I looked closer at the top genes that came up in our 1,582 gene signature, so most of the 100 genes that were popping up in our analysis, I've never heard about before. So I entered them multiple times to PubMed, and I then encountered the fact that a publication by Ron Crystal popped up multiple times, and that publication showed the transcriptome of basal cells, of airway basal cells. And that was the starting point. So the airway basal cells are highlighted in green in this cartoon, and they are the progenitor cell, the stem cell of the airway epithelium, and they sit on the basement membrane, and they give rise to any type of airway epithelial cell, so ciliated cell or globular cell or secretory cell. So they build up the airway epithelium. And at that time, so that was, I think, in 2011, Naftali said we should do it by ourselves, so we should not trust Ron Crystal's signature, so we should do it. And so we established in our lab all the methods how to isolate these cells, and we did transcriptomes of the cells. So we did transcriptome, obtained the transcriptome of macrophages, alveolar macrophages, of the basal cells, and of airway epithelial cells. And we integrated this, and we came up with our own signature of airway basal cells, which is very close to the one that Ron Crystal published. So we confirmed the Ron Crystal data. And if we used these genes, or our signature of basal cells, and imputed this into our data set of the BAL cells, you can see if we only look at the genes expressed by the airway basal cells, that also the airway basal cell genes exclusively, that they highly predict mortality in IPF. And I can tell you that most of these genes are upregulated in the BAL of IPF patients if they early die. So, of course, we got interested. So before, nobody has published anything about basal cells in the BAL of IPF or other disease types, and we found them. So they have specific markers, so you can stain them for cytokeratin 5. They also express often cytokeratin 6, and they have a transcriptional factor which is called P63, delta P63. And as you can see, obviously the number of basal cells is upregulated in the BAL of IPF patients. And we also, of course, got interested in where to find them in tissues. And I would like to note that we are not the first showing that there is a link between IPF and basal cells. So already more than 18 years ago, a pathologist called Marco Chilosi from Verona, Italy, showed that the epithelial cell layer that is covering the myofibroblast foci express markers of these airway basal cells. So they showed the delta NP63 transcription factor that is highly expressed and exclusively expressed by basal cells. So we confirmed the results of Marco Chilosi, and I would like to show you some of these very interesting immunohistochemistry data. Of course, you know that we have bronchialization going on in IPF tissues, and here the purple cells are all airway epithelial cells that you can see in this, surgical lung biopsy of a early patient. And if you look closer, you can see that the airway epithelial cells are replacing the type 2 cells. And sometimes in early patients, that can look really nicely organized and normally organized, the epithelium. But sometimes you also see these quite aberrant proliferation, or at least it looks quite bizarre and more tumor-like. We confirmed the Chilosi results that the fibroblast foci are indeed covered by cells which express airway basal cells markers like cytokinin 5 and 6 and delta NP63. And we often observed, and that is, I think, really telling. So here you can see clusters of airway basal cells within the interstitium, within the fibroblast or the extracellular matrix. And these cluster of cells, of basal cells, they appear to become hollow. And out of these hollow becoming airway basal cell clusters, you have the impression that the honeycomb cyst is evolving. So on the right figure, you see the honeycomb cysts, and they appear to evolve out of these cluster and strands of airway basal cells. And as already mentioned before, so single-cell data also support this, that there is an accumulation of airway basal cells in IPF tissues. So this is the Witzit group paper showing that the type 2 cells and the type 1 cells are replaced by basal cells and other airway epithelial cell types. And also Naftali's data showed, and he said that he presented this yesterday, that the type 2 cells are nearly gone in IPF tissues and replaced by bronchial epithelial cells. So these single-cell RNA-seq data perfectly fit to our observation in the BAL. And we think that in the next couple of slides, I would like to speculate about these findings. So we think that this is a process mimicking mechanisms which are going on in organ development. So we think that this is a misled recapitulation of developmental programs of lung development. And the starting point in lung development is that the airway basal cells, which are shown here in this publication in green, so that they build up strands, so they, like in the tissue of IPF, so they start as a cluster of cells, they build up strands, then the strands become hollow, and then we have this branching and tubing morphogenesis. So they build up tubes, and then they branch, and after 14 days, so at that time, the airway tree is developed, and only at that time then the development of the alveolar structure is starting. So the basal cell proliferation and the airway tree formation is the starting point of organ lung development. And this is interestingly not only true for the lung, it is also present in all other glandular organs, like the kidneys, like the mammalian gland. All glandular structures are built first by building up the tree, the ducts, and then at a later time point, the tissue, the parenchyma, is evolving. And with that, I would like to end and wrap my talk up. So the BAL cell gene expression profiling revealed that there's an unexpected airway cell-derived signature in the BAL that is strongly associated with IPF mortality, and that this probably resembles an aberrant recapitulation of developmental processes, and that this, from our perspective, may be one key driver of IPF and really reconstruction and leading to a deconstruction of the alveolar tissue. Thank you very much.
Thank you.